Kai from Chongqing University, Department of Forestry. And today my talk is evaluating the spatial extrapolation by two types of species distribution models in forests of central Taiwan. My outline is from Introduction to Conclusions for part. It's common to see species distribution models, extrapolations in recent studies. That means extrapolation have become more and more important and popular. Species distribution can provide the species measure of as a measure of species occupancy, which ecological surveys cannot cover. And fortunately, because of the combination of the field of remote sensing and geographical information system and statistical algorithm improvement, we describe ecological patterns of species, or we predict the ecological species can be enhanced over broader scale, broader spatial scale. Species distributions models can be divided into two types. One is SPSO, means the models only need species present sample only. And the other is SPAS. It means the model construction only need species presents absence sample or pseudo absence sample. In our study, we chose the model of SPSO. We use file-clean domain, and SPAS we use discriminant analysis, DA, and logistic multiple regression, LMR, to predict our species habitat. These are our materials and methods. Our study area is in Huishan Experimental Forestation, and the station is in irregular shape. So in order to easily compute our result, we sample the region to a rectangular region. The total area is 70,136 hectares, and the Huishan area occupied 7,477 hectares. And this is our study species. Red striped rhododendron is one kind of sandy cluster tree species. It always forms pure forest. It also is endemic species in Taiwan Central Mountains. Our objectives are composed of four parts. We aim at evaluating the overall performance of SPAS and SPSO which model we chose. Bioclean, domain, DA, and LMR. Which one is most useful for predict our species? Let's try for the danger. And the third is examining the ability to do spatial extrapolation with our chosen four models. And that is assessing the effect of different variable combination. This is our study process. We first do data collection. We produce red triangle danger sample data point layer. How do we collect? We collect it in situ RSR samples by GPS and laser range finders. And then we perform a post-process differential correction that will make our data have an accuracy of some figures. And so far, there are 118 samples collected from Tongfeng watershed in Huisun, and 61 samples from Guangdao watershed in the west of Huisun area. And next we collected digital elevation model, orthophoto maps, and two-stage spot-by images. We use these three images to produce elevations, slope, aspect, terrain, position, and vegetation index to meet by variable layers. And we overlay the red stripe photodendron sample data point and our five variable layers to clips. Clips the point of the five variable layers lying at the same position of our species point, of data point. And we can obtain non-suitable area and suitable area of our species. The next is experimental design. And one of our goals, we want to examine the ability to do spatial extrapolation with our four models. So we would evaluate the improvements of species distribution models based on different two test data sets. This is a full Tongfeng data. We make two experimental designs. And the first is we split the full Tongfeng data set to 2/3 and for training models, and remain 1/3 for validation. The other experimental design is use the same two-thirds of Tongfeng data set for training, and validation use for a total Guangdao data set. So the data number is for training. The 39. Next, we will do model construction. We will use these four models: outcome, domain, DA, in order to produce reliable results, we will repeat our data chain. Like what I used to say, we select 2/3 Donghong data randomly. We will repeat this position and repeat this process for 3 times. And we will compare the accuracy of the 4 models to determine the optical one. The model validation can be done in 2 approaches. One is split sample validation. Other is kappa. Use kappa as the accuracy basis. The split model validation means for each model, we will compare the prediction and the observation result and calculate the difference between them. We calculate the error rate. And the kappa, it is accuracy index, model accuracy index. It ranges from -1 to 1. And as the value increase, represent the model perform well. This is our result. And the number here is our variable combination. As we see the 4 models of the Quantile training, Quantile validation, the domain, the kappa value of domain perform the best of the 4 models. And compared to SPSO and SPAS, we see the mean kappa. This shows that SPSO and SPAS are no not significant different. And next we see the ED1 and the kappa of ED1 and ED2. The kappa value of 4 models, no matter often domain DA and LFR. The ED2, the kappa value of ED2 are less than ED1, that of ED1. This means we use Donghong based model to extrapolate Quantile watershed are failed. This table shows that outcome and DA most suitable habitat for red striped rhododendron. These are, and in sum, we have 6 conclusions of this study. The first is there are no significant